Hi, everybody. Thank you for coming to this Alliance of Genome Resources Office Hours webinar. Today, we're going to have Kimberly Van Auken give us an overview of Textpresso, full text literature search engine at the Alliance of Genome Resources. Kimberly, thank you very much for presenting, and the floor is yours.
Okay, great. Thank you, Carol, and thank you everybody for joining us on our Thursday Alliance webinar series. So I'm going to tell you today about one of the tools that we have at the Alliance, which is called Textpresso. And as Carol said, this is a full text literature search engine that we have and are furthering development at the Alliance. So let me just go back. So let me just give you an overview of what you'll learn today. So I will very briefly give another introduction to what the Alliance of Genome Resources is. I'll then tell you more about what Textpresso is, give you a glimpse behind the scenes of how it works, and then go through a couple of search examples to show how you can effectively search a literature corpus with Textpresso. And then we'll have time at the end for questions and answers. So just as a reminder, so the Alliance of Genome Resources is a coalition of model organism databases and the Gene Ontology Consortium that have come together to share common infrastructure and tools and provide our users with a consistent experience in navigating a functional and genomic database across these model organisms. So the current members of the Alliance are Flybase, Mouse Genome Database, Rat Genome Database, the Saccharomyces Genome Database, Wormbase, Zenbase, ZFin, and I said also we're joined by the Gene Ontology Consortium. So many of you, if you have been to the Alliance, you're probably most familiar with the Alliance Gene pages, and this is one example of those. And so this is the portal where our users can find in-depth gene function information across multiple species. This is just an example of the mouse NLRP3 gene. And over here on the side is all the different kind of information that we currently have at the Alliance about genes in these various model organisms. And I'll particularly point out the orthology section as one of the places where it's really nice now to do comparative genomics. But in addition to these gene pages that you see, the Alliance is also working hard to develop a number of tools that our users can use to help them with their analyses on the biology of these various model organisms. And so the tool that I'm going to talk to you about today is Textpresso. And I will walk you through some searches and some of the features of Textpresso if you want to follow along in real time. If you go to the Alliance of Genome Resources webpage, alliancegenome.org, under Data and Tools, you'll find a link to a Textpresso page. And if you click on that link, you'll be taken here. And it will give you a brief overview of what Textpresso is, and I'll be covering that. And then there is a list of the current Textpresso instances that we have for MGI, SGD, Wormbase, and ZFin. So what is Textpresso? So here are the key features of Textpresso as a literature search engine. So like I said in the beginning, Textpresso allows you to search the full text of the literature that we have. So not just the abstract or the title, but the actual full text. Another key feature of this, and this is one of the nice things about having this integrated into the Alliance, is that the bibliography or the papers that are included in Textpresso are papers that have been selected or curated by the given model organism database. You can use Textpresso to perform simple keyword searches, but the real power of Textpresso, and I will demonstrate this, is that you can perform advanced searches using what we call categories, which may be ontologies that you're already familiar with, like Go or the disease ontologies, but also includes categories that have been created by curators to find information about specific topics. And these are groups of words that curators have determined that help describe particular kinds of experiments. So they can be very useful for performing particularly targeted searches, and I'll show how to do that. The results of the Textpresso searches are presented in the context of the matching text. So you can always see exactly what the search engine found in the paper and why it thinks it's relevant. And then you can actually go out and see that in the context of the paper. And then also the results, you can sort by year, you can put various filters on your searches and download those search results. So, like I said in the beginning, one of the great features of the Alliance of Genome Resources is that we are now able to use common infrastructure and software and tools for the various model organism database members. And so we are, in addition to the public-facing web pages that you see, we have a whole backend infrastructure, and part of that backend infrastructure is our Alliance literature system. And this has enabled all of the groups to begin to model and then eventually adopt Alliance infrastructure for bringing in papers to their bibliographies. And this then has enabled us to create these Textpresso implementations for the different mods at the Alliance. So currently we have them for MGI, SGD, Wormbase, and ZFIN. There are slight differences in both the current corpus that's available and the kinds of these categories or ontologies that are available. All the implementations have access to the text that's available as PubMed Central Open Access. But in addition, both SGD and Wormbase have included their entire PDF collection of references that they've included in the SGD and Wormbase bibliographies, respectively. All of the implementations have as ontologies available for search. Like I said, some of the more commonly used ontologies in biological curation. So KEBI, the disease ontology, Go, and sequence ontology. In addition, to maximize the effectiveness of the searches, we also are including some of the organism-specific categories or ontologies. So certainly things like gene names, proteins, if the syntax is different. We may have things like anatomy ontologies, life stage ontologies, lists of alleles, strains, variations, phenotypes, and for the ZFIN implementation, things like fish and cell types. And then also, as I mentioned, particularly for the worm-based implementation, we have a number of curation-specific categories that we've created that are very helpful for finding particular kinds of information in the literature. So what I'm going to do from this point on is really show some of the specifics of how to perform searches, and I'm going to do that with the implementation that we have for C. elegans, since I've been a worm-based curator for some time now, and this is the one that we use most often, and our community also uses as well. So here's the homepage for the Textpresso for C. elegans literature. And what I'm going to focus on for the talk here is really the search implementation. So you can access search in a couple of different ways. If you want to just perform a keyword search, you can enter a word or phrase in this window as well as down here. But if you want to perform an advanced search, and that's typically what I do when I'm using Textpresso, you can either click on the search tab at the top or the advanced search here. So if you click on that, this is the interface that you will come to. So I'll point out a couple of the main features of this. So when you're using Textpresso for search, you have the opportunity to select what the scope of that search might be. There are two choices here. You can either search through the whole document, or you can specifically search within a sentence. And I'll show how that becomes relevant later. And then you can also, where we're able to separate documents into their sections, you can pick a particular section of the paper. Maybe you just want to look at the results. Maybe you, for some reason, just want to go through abstracts, or maybe you're looking for a reagent that might be in the methods. This is where you would type in any keywords for your searches, and that can be a case-sensitive search. If you want to also include some of the categories in your search, this is where you access the menu of categories. And then lastly, I've just opened this up here. There are additional search options. This is where you can perform the filtering and the sort by year, where you can exclude keywords, look for particular authors, journals, the year that something might have been published, a particular accession, or a type of paper. Maybe you really just are trying to find something in a published experimental article, say, and not a review. So now we'll take a little bit of a glimpse behind the scenes. I think it's helpful from using Textpresso to really get a sense of exactly how it works. So what happens is we take the full text of the corpus for a given mod, and this is typically PDFs, and using our software that gets converted to text. Now, I will just note that that is not a perfect process. Sometimes there are issues with converting the PDF to text, but for the most part, I think it gets us very far. We then take that text, and it is indexed. The full text is indexed for keyword searches. So if you do your keyword search, you can find that anywhere in the full text. But what it also does is it takes that text, and where we either have an ontology or a list of entities like genes or particularly semantically related words, Textpresso goes through and actually tags those words so that it knows, the software knows, what meaning there is to that particular word. So here's an example. It might know for the C. elegans literature that all of these capitalized versions of these gene names were actually there referring to C. elegans proteins. Or list of terms like nucleus, plasma membrane, endoplasmic reticulum. Those are subcellular components. And if you were, say, interested in the localization of a particular protein, we have categories that have words like accumulates or localized, resides. So Textpresso knows that this category of localization are all the kinds of words that might describe where a protein is localized. And this makes searching for particular kinds of information much easier, because you might know generally what kind of question you're asking, where is this protein localized, but you don't want to necessarily have to try to think of all the different ways this might be described in the natural language of papers. We've already done that for you. So this again just illustrates the markup process with a recently published C. elegans paper. So here's the text from the paper that's describing the experiment that you see here. So I've taken this sentence out from the text. It starts with examination, refers to the figure, and this is blown up here. And this is just illustrative of what's happening behind the scenes with Textpresso. And what I've done is color code the different words and phrases that are in this sentence that are then classified by the Textpresso software. So things like examination and levels might be terms that are indicative of an expression experiment. And we said the protein name, SBP1, is marked up. AC actually refers to a particular type of cell in C. elegans. The pink terms are verbs that describe these experiments. L2, L3 molt, one cell stage are from our life stages included, and there are others in there as well. And we've even included a category that refers to figures and tables. So if you really want to find sentences in the literature that refer to a particular figure or table that are most likely to describe an experimental result, that category is available as well. So then, given that and a little glimpse of how it works behind the scenes, let's go through some examples. So like I said, Textpresso, it can work very well for just doing simple keyword searches. In fact, it works sometimes almost too well. But what I'll show here is, say you're interested in this particular protein, SBP1, and you want to know what are recent papers that have experiments about SBP1. So you can type that into the keyword search, do case sensitive. I'm going to sort by year. And then this is what the results will look like when they're returned. So the Textpresso search, that takes maybe about 2 to 3 seconds, found 49 documents. And this is probably much more than you will get necessarily in performing a search in PubMed. But the nice thing about the Textpresso results is that in addition to the basic bibliographic information that you're seeing here, the title of the paper, PMID, where it was published, the kind of paper it is, you'll get a score. You'll get a numerical score that's based on a term frequency, inverse document frequency calculation. And it will tell you, based on the information content of what was returned by that search, of how relevant these particular papers are to your query, right? So these are sorted by year, but I can sort of quickly go through the list and say, okay, of the papers that were most recently published, here are the ones with the highest score in the list that are most likely to have significant information about SBP1. And you can certainly, of course, look through all of the results that you get and decide what's relevant. And so what do you do with those results? So if you open up the arrow for each of the matching papers, what you will actually get from Textpresso is the text in the full text that matched your search query. And so I'm just showing you a sample of the sentences that are returned, but you can go through and scan through and say, okay, this is what Textpresso found that says that this paper is relevant. Right? And further, what you can do is you can actually check on this box and then view the results in the context of the full text here. And so this is what this looks like. You can scroll through the matching sentences. They're highlighted in yellow. You can then see things and your results with, say, the figures, and then get a good sense of what kind of information does this paper have, and is it relevant for what I'm looking for. So Textpresso works very well as a keyword search engine. If there are examples of your keyword in a paper, it will most likely find it. And like I said, what I think the real power of Textpresso lies is being able to search for specific kinds of information or specific facts, as we refer to them. So this is what I'm going to show you an example of how to do here. So let's go back to our protein SBP1. And what I am particularly interested in, say, is finding papers that describe its subcellular localization. And to do that, I'm going to ask Textpresso to find sentences that have at least one match to particular categories that relate to expression experiments. So for the search scope that I've done here, I'm going to ask Textpresso, look for all of these things in a single sentence. Look for the protein, so that's a case sensitive. And then if we open up this category menu, You will see for each of the implementations, all the basic ontologies, but then for Wormbase in particular, if you scroll down, you can open up a menu for what we refer to as the Wormbase curation categories. And these are often the topic-specific categories that curators at Wormbase have developed, and they are very helpful for finding specific pieces of information. So in this example for finding subcellular localization, we have a group of categories underneath the cellular component for Go. That's one area where we curate subcellular localization. And I picked each one of these categories. So assay terms for experimental expression assays, the actual cellular components as they're typically found in the literature, and then what we refer to as the verbs that describe these experiments. And I've selected each one of those, and I've told Textpresso I want to find sentences that have matches in a sentence for all of these categories. And you can choose at least one or all. So I picked those. And then these are the results that you got. And so again, if you open up a particular paper that's returned, it will show you the matching sentences from the full text. And in this case, you'll see a score for each of these sentences and which ones had the highest matching score for the search criteria that I sent. And you'll see that it's marked up, as I showed you earlier, for each of the particular categories that I asked it. And so you can see exactly what the sentences are that Textpresso found that are describing the experiments that show the nuclear localization in the anchor cell as published in this paper. And then again, if you want, you can go over, view the selected paper, open that up, scroll through your results, and find in the context of the full text where these sentences actually lie and what experiments they refer to in the paper. And so another example of where this might be useful is say you're interested in exploring the genetic interactions for the SBP1 gene. In this case, I've done a similar search where I've asked for to return sentences that have matches to the categories. I'm looking for the SBP1 gene, so that's a case sensitive for lowercase. And then I'm going to ask Textpresso, in those sentences that mention SBP1, look for another C. elegans named gene, so indicative that there might be a description of a genetic interaction. And then we have a category of phrases that are relevant for genetic interactions. So I've asked it to find sentences with all three. And here again are the kind of results that you can get, where each sentence is marked up. You can see where it's talking about suppression and synthetic lethality. And again, you can go to the full text in the paper, find these matching sentences while you scroll through, look at the accompanying figures, and get directly in the paper the kind of information that's describing SBP1 genetic interactions. So the hope, of course, is that in our model organism databases, we would have all of this information already curated, but many of us also have curation backlogs. So in the interim, Textpresso is a great tool for being able to find information. And if you find things that we don't yet have curated, we would, of course, love to hear from you so that we can add them to the database. So the last thing I will mention as far as Textpresso is, I've talked a lot about these particular categories. If you're interested in exploring more of the content of those categories, on the tab at the top of the page, you can go to Browsers, and you have two options. You can either pick a particular category from this tree and browse through the terms that are in that category. Or if you want to perform a search, you're looking for a particular kind of information, but maybe you're not certain which of these categories should I select in my search. You can search for terms, and then it will show you the matching categories for that search term. And so for this example, I've typed in suppression. And so you can see that there are a couple of categories that actually where that word is used. But if you were interested in genetic interactions, it will show you that that word is contained in our genetic interaction category, and you can use that. But you can see there are other things that have that word, maybe some of the phenotypes in the worm-based phenotype ontology, or the disease ontology, for example. But that's a good starting point to start to familiarize yourself with the content of the categories and which ones might be particularly useful for searches you would like to do. So that is basically a quick overview of how to do meaningful and hopefully effective Textpresso searches. I will be happy to take questions at the end. Also, I just wanted to close by reminding everybody that there are upcoming Alliance webinars. So if you go to the Alliance homepage under news, there's an event calendar that lists the upcoming webinars. Another webinar that we will be giving in April will have more to do with how the Alliance is embracing state-of-the-art artificial intelligence to augment our literature curation as well. So I would urge people, if that's something that interests you, you can come hear more about how we're making use of our common literature infrastructure and some of the really exciting and fast-moving AI that's out to help inform and improve our literature curation. And so lastly, as I said, the Alliance is a consortium, and we brought together a lot of great people to help with software development and curation. So I want to especially thank some of our software team: Valeria Arnabaldi, Juan Carlos Chan, Hans Michael Mueller, Carrie Van Slyke, who is one of our product owners, Daniel Wang at Wormbase, who makes sure that we have all the PDFs for Textpresso, and Shuai Yang. Also, thank the Alliance web team for making Textpresso findable for people. And lastly, two of the Alliance PIs, in particular, Carol Bolt and Paul Sternberg, for their support for the webinar. If you have any questions at all, feel free to get in touch with us at help@alliancegenome.org. I've also listed here the GitHub repository for the Textpresso code. And if you're interested in learning more about Textpresso, there is also a paper published several years ago in VMC Bioinformatics. So again, thanks everybody for attending. And if you have any questions, we'll be happy to take them.
Thank you, Kimberly. And I believe that everyone now has the ability to unmute themselves if you'd like to ask a question, or if the chat is open, you can type something in the chat, or I believe we have the Q&A dialog box open, and you can use that as well. So Kimberly, I had a question to start with, which is,
Sure.
If I go to Textpresso right now and I search the mouse,
okay,
and when I look at the papers that come back in response to my keyword searches, they all seem to have MGI IDs, which means that I think they've been curated by MGI, but I don't know for what categories. Is there thought on, when you get the results back from your keyword, if there's a paper that the mouse team or the yeast team or the worm team has already annotated for certain concepts like gene or function, or is there the ability to sort of see that in the search results, that yes, we've looked at this paper, here are the things that we curated it for, or do you have to follow the links and go back to the model organism database itself to find that information?
Yeah, so that's a great question, Carol. And I think that That is something I would love to see us be able to do, is to integrate Textpresso search results when one is already been curated or is known about that paper in the context of the Alliance or the particular model organism database. We don't have that yet, so you would have to follow the links. But yeah, I think that's something that would be great to do.
And are there like near-term enhancements that are being planned for Textpresso that you can comment on?
So as I was getting ready for this talk, I certainly have a list of some things I think that we could do. I think that, improving the content of some of the sites that currently maybe only have PMC open access, because we know that there is other literature available. I think that would really help improve the searches, as well as extending the categories that, say, I showed for the worm-based implementation that could be equally useful for any other, I don't know, organism as well. So it'd be nice to be able to do that. And then, as you mentioned, I think the other thing that would be really helpful is to link what we might already know from those papers to Textpresso, so that users can more easily navigate between their search results and the actual Alliance, say, gene pages with the curated data. But we're certainly open to other suggestions as well, if there are things that people would find particularly useful.
Great. There is a question.
Mhm.
Are terms from the sequence ontology searchable in Textpresso? For example, ZFIN's literature corpus, and if so, what do the results look like?
So the sequence ontology is included in our categories. I haven't actually performed a search of the current ZFIN literature with the sequence ontology, so I can't speak to that. I don't know if any of the ZFIN curators who might be on the call have done that. Just one thing I will, just sort of as a note of caution, is that as incredibly useful as ontologies are for biocuration, I think we all know that authors don't always necessarily use those terms when they're writing in natural language. So I think sometimes the ontology searches of the literature can get you so far, but are maybe not as precise at finding natural language as we would like. That's partly why we developed some of those worm-based specific categories. But yeah, if any of the ZFIN curators are on the call and could speak to that, that would be great.
I think Michael was trying to unmute yourself. Michael, try to unmute yourself now.
Yeah, I can. Yeah, I can. So, with respect to enhancement in Textpresso, the previous question. So we are also working on now implementing AI-powered features. So document classification, sentence classification via neural networks, right? So that we can later on search, let's say, sentences and documents that are important for certain classifications, like is this a paper that contains interaction, or is this a sentence that describes interaction? Right, so similar to what the category searches is, but just we want to do that, use AI technologies, neural networks, and LLMs to enhance the search quality, basically.
Thank you, Michael. And Karen Christie, you were asking about an MGI.
Yeah,
version.
so I noticed that Kimberly mentioned that there is an MGI version, but it was specific to PubMed Central, and MGI's curated corpus of PDFs. Sorry, I probably should have used the word PDF. Oh, maybe I did. That we've curated our our corpus of PDFs broader than just what's in PubMed Central. And that's, I was wondering, and at SGD, when I was still there, we started using Textpresso, and it can be really useful for searching and finding the papers that you really want. So I was wondering, and I noticed that I think it was ZFin doesn't have their PDF corpus included yet either. So I was wondering if there were plans to add to those Textpresso instances, the PDF corpuses that are curated by those respective databases.
Yeah, I don't know if one of the developers wants to comment further on this, but I think it's something that we would love to be able to do. Because you're right, it makes it that much more useful, right? When you have access to the largest corpus you can possibly search.
It is on the roadmap for Mouse. It just hasn't been implemented yet.
Hi, Kimberly. I had a question.
Sure.
So I was struck by the thought that we could visualize the search results in the context of the paper. I really hadn't paid attention to that feature, though I'm a regular user of Textpresso. And it struck me that it might be really useful for curation assistance, because we're talking about how we can improve efficiency of curation, and we're talking about AI methods and stuff in the AI working group at the Alliance. But I was thinking if this could be plugged into the curation tool, it's already quite efficient in pointing out what section of the paper, what sentences right away can give me clues to, you know, the terms that I would put into my curation form. And of course, we could, you know, definitely use AI and explore AI methodologies. But before we get there, we already have this. And if that could be plugged in, you know, just integrated more closely to our curation forms, that would be really great. And I think we had this idea, I think, in Wormbase years ago, where we would open the paper, Textpresso would highlight all the entities, and somehow this would be integrated very closely to a curation form. And we could even click on entities and categories and search results in the index text, and it would automatically fill up.
Yeah, absolutely.
I mean, we had all these ideas in the past. Yeah.
Yeah, I think you're spot on, right? I think this is also another way that we could use the search results and integrate it more closely with our curation tools. I don't know how many people here on the call. There's a gene ontology curation tool called Noctua, and a few years back we piloted exactly that kind of thing with helping perform Go curation in Noctua. And so, yeah, I think that would be another fantastic thing to do, both for professional curators as well as for, you know, our great community curators, to help them to help us to get information into the databases more quickly.
Yeah. Great. Thanks.
Thanks, Ranjan. Any other questions or comments from folks? If not, Kimberly, I want to thank you again for great presentation, and for everyone for their questions and comments. The recording will be posted to the Alliance website. So if you missed part of the presentation, you can go back and look at the recording from the very beginning. And we will have our next Alliance office hours. I believe it's February 20th, and Jennifer Smith is going to be talking about variants at the Alliance. So thank you all for coming, and hope to see you again at the next office hours.
Great.